I want you to just take a second and imagine your immune system as this vast, ancient library.
Right, a really old one.
Yeah, exactly. And it's filled with literally millions of blueprints. And your body is just constantly scouring these blueprints to build weapons, antibodies basically, to fight off viruses and bacteria that it has never even seen before. It's an absolute marvel of evolution.
Oh, totally.
But what if I told you that this ancient library is fundamentally limited by the very architecture of how it was built, like millions of years ago?
Yeah.
Because the shelves are physically bolted to the floor, the wings are fixed in place, and honestly, half the books on the shelves are just blank pages taking up space.
Yeah, it's a profound structural limitation, really, because biology is incredibly inventive, obviously. But it's entirely bound by the physical linear constraints of the DNA string it just inherited from previous generations.
Exactly. But what if we could bypass that? Like, what if we could just burn down that ancient disorganized library and build a biological supercomputer in its place?
That is the dream right there.
All right, so today we are doing a deep dive into how a team of scientists did exactly that. We're unpacking a truly groundbreaking 2026 paper from Cell Genomics by Fu and colleagues. And the mission of this deep dive is to explore how we're not just observing the human immune system anymore. We are talking about scientists who literally wrote an entirely new human immune system from scratch, pasted it into a living mouse, and then program that DNA to shuffle its own sequence on command.
Yeah. The sheer scale of this achievement is, frankly, it's difficult to overstate. I mean, we are looking at an absolute masterclass in synthetic biology here.
It's just wild.
It is, because for decades, genetics has been, constrained by natural evolutionary limits. We could read DNA, and eventually, sure, we could edit small pieces of it, like with CRISPR. But this paper, this represents the leap to programmable, large-scale genomic rewriting inside a living, breathing mammal.
Okay, so to really grasp how they rewrote the code, I feel like we first need to understand the hardware they were trying to replace, right?
Yeah, we have to start at the foundation.
So that brings us to the natural immune system's core engine, which is a mechanism called VGJ recombination. Can you walk us through that?
Absolutely. So VGJ recombination is essentially how your body generates diversity in its antibodies. Your natural immunoglobulin heavy chain locus, which let's just call it the IGH locus for sanity.
Yeah, please. IGH locus it is.
Right. So the IGH locus is a massive continuous stretch of DNA. It is about 1.2 megabases long, which in genetic terms is huge.
Huge.
And scattered across this massive string are these discrete genetic segments. They're classified as V for variable, D for diversity, and J for joining.
Okay, let's unpack this for a second, because I've heard this described sort of like a genetic slot machine. Like you pull the lever, the wheel spin, and you randomly get one V, one D, and one J. Boom, they snap together, and you've just built the blueprint for a specific antibody to fight a specific virus.
That's a super common analogy, yeah.
But wait, I love that analogy, but if the DNA is just a physical string sitting inside the nucleus of a B cell, how do the reels actually spin? Aren't they stuck exactly where they are on the string?
And that is the exact flaw in the slot machine analogy, and it honestly highlights the central problem these scientists wanted to solve. The reels don't spin freely at all.
They don't?
Nope. During immune cell development, there's an enzyme called RAG that acts as the architect. RAG binds to a J segment, and then it physically pulls the upstream DNA through itself, creating this massive loop.
Oh, wow. So it's actively pulling it.
Yes. It linearly scans the physical string until it grabs a D segment, then it cuts the DNA and pastes them together, and then it literally repeats the entire process to go up and grab a V segment.
Okay, so it's not a slot machine. It's more like pulling a really long strip of magnetic tape out of a cassette.
Precisely. That's a much better way to look at it. And because it is a physical tape, the spatial positions of these V, D, and J segments strictly determine how likely they are to actually be used.
So location matters.
It matters entirely. If a V segment is located right next to the D segments, it gets picked all the time. But if it is, a million base pairs away at the far end of the tape, it rarely gets used at all.
Not to mention the tape itself is kind of a mess, right? Like naturally.
Oh, total mess. Natural human populations carry widespread structural variations. So depending on who your parents are, you might have inherited large-scale deletions, which means you are completely missing entire functional genes.
Wait, really? just missing them entirely.
Yeah, just gone. Or, on the flip side, you might have non-functional pseudogenes, which are basically evolutionary leftovers, just acting as junk DNA.
Just cluttering up the tape.
Exactly. And because that RAG enzyme has to linearly scan past all this junk, the physical structure places a hard limit on the diversity of antibodies your natural body can actually produce.
Okay, so just to make sure I am totally tracking here, and for everyone listening, let's do a quick recap. The natural immune system relies on this incredible cut-and-paste mechanism to mix and match antibody parts. But because it relies on a physical, rigid strand of DNA that's filled with junk and missing pieces, our bodies are, like, artificially capped in how many unique defenses they can mount.
That's a perfect summary, yes.
Which makes me wonder, as someone listening to this right now, when you think about the DNA you inherited from your parents, you just assume you're stuck with it. But if you could rip out that old cassette tape and replace it, what kind of modern diseases would you want it optimized to target?
Right, it really changes how you think about your own biology. Because if the natural locus is physically constrained, the only way to decouple our immune diversity from those limits is to just completely rewrite the code from the ground up.
And that is exactly where Fu and the team come in. I mean, they didn't just go in with CRISPR and snip out a few bad genes. They engineered a brand new, completely synthetic locus called SynIgh.
Right, synthetic IGH.
But my question is, how do you even start building something like that? I mean, how do you organize 1.2 million base pairs of messy DNA on a computer?
You do it by stripping it down to its absolute bare essentials. They took that bulky 1.2 megabase natural locus and they reduced it to a highly streamlined 174 kilobase synthetic version.
Wow, that is a massive compression.
Huge. They deleted every single pseudogene, all the junk DNA, and those massive unnecessary spaces between the genes, and they built it using a strictly modular, bottom-up design.
Okay, when you say modular, do you mean they treated the genes almost like standardized software components?
Exactly like software. Every single V, D, and J gene was structured as a distinct, standardized module. For instance, a V unit isn't just the gene itself in this design.
What else is in it?
They packaged it with a starting signal, the actual coding sequence, and then an ending sequence called a recombination signal sequence, or RSS.
Okay, what did the RSS do?
It acts like a chemical cut here sign for that ARG enzyme we talked about earlier. So every module is perfectly formatted. They even standardized the regulatory regions. They took massively long natural control sequences and engineered concise synthetic versions that do the exact same job in a fraction of the space.
So they have this beautiful, clean, totally streamlined framework, but you still need to populate it with actual genes, right?
Yeah.
Like the actual books for the library. If they aren't just copying one really healthy person's DNA, where do the actual antibody blueprints even come from?
Oh, this is where the project scales up dramatically. To make the SynIgh locus better than any single natural human's DNA, they analyzed global databases.
Global databases, like across the world.
Yes, they used the 1000 Genomes Project and international immunogenetics databases to find the most frequently functional genes across diverse global human populations.
Wait, so they essentially crowdsourced the immune system.
They absolutely did. They selected 60 V genes, 31 D genes, and 13 J genes that literally represent the pinnacle of human immune function globally.
It is mind-blowing.
They packed the best human immune components from all over the world into one single synthetic sequence.
Greatest hits compilation of human immunity. But, okay, we established earlier that the biggest flaw with natural DNA is that it is rigid, right? Like a cassette tape.
Yeah.
So even if you pack it with the best genes in the world, it's still a static tape. How did they fix that fundamental hardware problem?
By introducing a system they called mSCRaMbLE. It stands for Mammalian Synthetic Chromosome Rearrangement and Modification by LOXP-Mediated Evolution.
Yeah, okay, let's definitely ignore that acronym immediately and just talk about what it actually does.
Fair enough. It's a mouthful. Basically, they wanted the DNA to be able to rewire itself. So when they were designing those standardized V, D, and J modules on the computer, they physically inserted specific genetic hinges called LOXP and LOXIM sites between every single gene.
Okay, genetic hinges. I like that.
Yeah, think of these hinges as docking stations for a specific pair of molecular scissors. Later on, if those scissors are introduced into the cell, they will lock onto these hinges and instantly cut, invert, and shuffle the DNA into completely new structural variants.
Okay, let me pause and synthesize this real quick because the engineering here is just wild. Let's do another mini recap. Scientists took our sprawling, cluttered 1.2 million letter immune DNA and compressed it into a sleek 174 kilobase synthetic drive. They threw out the junk, crowdsourced the 104 best human genes from global populations, and then, just to show off, basically, they installed chemical hinges between every single gene so the whole thing could eventually shuffle itself.
That is exactly what they did.
It really makes you think, as a listener, how would it feel knowing your body was running on globally optimized, crowdsourced genetic code rather than just the random genetic hand you were dealt at birth?
It's a profound shift in how we even view biology. But, designing this beautiful, streamlined sequence on a computer screen, that's honestly the easy part.
Like, you actually have to print it.
Exactly. The grand challenge of synthetic biology is actually synthesizing that DNA and assembling it in a physical lab.
And that brings us to a huge obstacle, right?
Yeah.
Because that modularity we talked about, you have 60 V genes that are incredibly similar to each other. I mean, the paper noted that some of their coding sequences are up to 99.6% identical.
Very close, yeah.
So you're trying to assemble this massive strand of DNA, and it's like trying to perfectly stack 100 identical slippery Lego bricks. But researchers usually use host cells like yeast or E. coli to assemble large DNA constructs, right? When you put all those identical Legos into a yeast cell, doesn't the cell's natural machinery just completely freak out?
It does. It totally freaks out. The host cell has its own DNA repair machinery, which constantly scans for errors. So when it sees all this highly repetitive, nearly identical synthetic DNA, it assumes a massive replication error has occurred.
It thinks it's a mutation.
Exactly. So a process called homologous recombination kicks in, and the cell's enzymes essentially chop out the repeating parts, smashing the sequence together and totally destroying the code.
Wow. So the very cell you are using as a factory to build your DNA is actively destroying it because the code is too perfectly repetitive. How do you stop the cell from ruining the sequence?
The researchers had to engineer a brilliant biological fix. They created a synthetic stabilizer protein. See, in nature, highly repetitive DNA, like the centromeres at the center of our chromosomes, is kept stable because it is bundled up so tightly that the cell's repair enzymes physically cannot reach it.
Oh, so they wanted to mimic that natural bundling. Okay, how?
They took DNA-binding proteins. So think of these as molecular anchors. Specifically, they used proteins called DCRE, POU5F1, and CTCF.
Okay, molecular anchors.
Right. And these anchors are designed to specifically grab onto those synthetic hinges we discussed earlier. And then they fuse these anchors with a very unique effector protein called HP1 alpha.
And what makes HP1 alpha so special?
HP1 alpha drives something called phase separation. The best way to visualize this is mixing oil and water.
Oh, like how they form little beads?
Exactly. HP1 alpha attaches to the synthetic DNA and causes it to condense into a dense, liquid-like droplet inside the cell. Mm-hmm. It basically acts like a biological vacuum sealer.
A vacuum sealer, I love that.
Yeah, it balls the massively long, repetitive synthetic DNA up so incredibly tightly that the cell's bulky recombination scissors simply bounce off the outside. They physically cannot fit inside the droplet to cut the repeating genes.
That is genius.
Yeah.
So it's not a chemical shield, it's a literal physical one. You are vacuum sealing the fragile DNA to protect it from the chaotic environment of the cell.
Yes, and the data on this is just striking. When they tried to assemble the DNA without this HP1 alpha stabilizer, they got 0 correct clones.
0?
0. The DNA just kept collapsing into garbage. But with the stabilizer turned on, they successfully assembled the full 174 kilobase synthetic construct. And they kept it stable in yeast for 30 days without a single accidental deletion.
30 days. It completely solved the assembly problem for highly repetitive mammalian DNA.
It did. It was a massive breakthrough just on its own.
Which is such a massive hurdle cleared.
Mm-hmm.
So to recap again, we went from a fragile, repetitive string of code that just kept destroying itself to using a custom vacuum sealer protein to perfectly assemble and protect it. And it makes you wonder, think about the fragile systems in your own life or work or technology. How often could we benefit from a mechanism that just compacts and protects our most vital information from the chaotic environment around it?
It's a great thought. But, having a perfect, stabilized string of DNA sitting in a yeast cell doesn't really do us much good in the end.
True. Yeast don't need antibodies.
Right. The ultimate test of this entire project is whether a living, breathing mammal's body will actually recognize, read, and utilize that artificial code.
Exactly. So they have to transition from the petri dish to a living organism. They took this 174 kilobase synthetic DNA along with something called piggyback transposase and injected it directly into fertilized mouse eggs. How does the piggyback part actually work?
So piggyback is essentially a highly efficient molecular copy and paste tool. When injected into the embryo, the piggyback enzyme grabs the ends of the large synthetic DNA sequence and smoothly pastes it right into the mouse's own genome.
Okay, so the mouse is born, it grows up, and it has this massive block of human-designed DNA sitting in its cells. How do you even check if the mouse's body is actually using it? I mean, did they just take a blood sample?
They actually went straight to the source, the bone marrow.
Oh, okay.
Bone marrow is the nursery where early B cells develop, and it's where that RG enzyme we talked about earlier does its cut and paste routine. The researchers used a highly sensitive sequencing technique called VDJ-SIFIC.
What does that do?
They basically created molecular baits that were specifically designed to only catch the human synthetic J genes. This allowed them to pull out and read exclusively the antibodies that were built using the artificial code, completely ignoring the mouse's natural immune system.
And the proof of function was absolute, right? I mean, the paper said they found 7,971 unique immune combinations.
Yes. And it wasn't just a handful of genes doing all the work either. The sequencing proved the mice perfectly utilized every single one of the 60 synthetic V genes, all 31 D genes, and all 13 J genes.
That's incredible.
The synthetic locus was responding perfectly to the mouse's natural developmental signals.
Wait, so just to be absolutely clear, the biological machinery of a mouse seamlessly read human-derived, computer-optimized DNA and built functioning immune cells out of it.
Seamlessly. And what is perhaps most impressive is the predictability of it. When they sequenced the bone marrow from several different mice carrying the SynIgh locus, they compared how often each specific gene was used across the different mice, and they found a Pearson correlation coefficient of 0.9.
Okay, for those who haven't looked at a statistics textbook recently, what does that 0.9 mean?
Right, so a correlation of 0.9 out of 1.0 means it is incredibly consistent. It proves that this synthetic design isn't just generating random biological chaos.
It's stable.
Exactly. It functions as a highly conserved, predictable system. It behaves exactly like a natural locus, but it is running entirely on our engineered code.
Let's just sit with that for a second for another quick recap. They pasted 174,000 base pairs of synthetic code into a mouse embryo, and that mouse grew up to successfully read those artificial human genes as if they were its own. It generated nearly 8,000 functioning immune combinations with mathematical predictability. I mean, imagine finding out a piece of your own bone marrow was running flawlessly on crowdsourced synthetic DNA. At that point, does the line between natural biology and artificial technology even matter to you?
It certainly blurs the line to the point of irrelevance, I'd say.
Totally.
But, here is where we push past simply mimicking nature. The synthetic locus works predictably, which is great. But the entire purpose of the SynIgh project, specifically those chemical hinges we discussed earlier, was to exceed natural limits.
Yes, the mSCRaMbLE system.
Right. It is finally time to activate that flexibility module and really shuffle the deck.
Okay, so they built the ultimate optimized slot machine. But now they want the machine to literally disassemble and rearrange its own reels while it is running inside the living mouse.
Exactly.
So to do this, they bred their synthetic mice with a specific strain of mice called RAG2 knockout mice. Why that specific breed?
Because a RAG2 knockout mouse lacks the RAG enzyme.
The cut and paste architect.
Right. Without RAG, the mouse cannot perform early natural VDJ recombination. So breeding them effectively hits the pause button on the mouse's early immune development.
Okay, so the system is paused. The B cells are just waiting in the bone marrow. What's the trigger? How do they unpause it and shuffle the DNA?
Within this pause state, the researchers inserted a highly controlled chemical switch. It's an ABA inducible split Cree system.
Okay, let's unpack the split Cree because this mechanism is fascinating. Cree is the molecular scissors that can lock onto those hinges we built into the DNA, but they split it in half.
Exactly. They took the instructions for the molecular scissors and literally split them into two inactive pieces. Both halves are just floating around inside the cell doing absolutely nothing.
Okay.
But each half is attached to a sensor. ABA, or abscisic acid, is a simple, harmless plant hormone. When the researchers inject the mouse with ABA, the chemical acts like a magnet.
Oh, I see where this is going.
Yeah, it binds to the sensors, instantly snapping the two halves of the scissors back together inside the living cell.
So by simply giving the mouse a harmless plant chemical for 5 days, they dynamically turned on the scissors.
Yes.
And those scissors went straight for the hinges, triggering the DNA to violently cut, invert, and shuffle its own genes.
Precisely. And the results of this are staggering.
Break it down for us.
Let's look at the D region first. In nature, RAG scanning is highly directional. It's like reading a book. It almost exclusively reads the D genes from left to right.
Right.
But when the split Cre scissors activated, they grabbed the hinges flanking the entire D region and physically flipped it backward.
They inverted it.
Yes. The researchers actually scanned the whole genome looking for translocations to prove this physical flip occurred, and found that over 17% of the cells successfully inverted their D region on command.
And that inversion basically unlocked an entirely hidden dimension of antibody architecture, right? Because when they sequenced the final antibodies, those inverted D genes, which are like almost non-existent in nature, suddenly made up 41% of all the immune products in the shuffled mice. They literally forced the enzyme to read the book backward.
They did, and the transformation in the V genes was just as radical. By activating the hinges between every single V gene, the region scrambled itself entirely.
So spatial rules are gone.
Completely gone. Genes that sat at the very end of the physical tape and were almost never used, like sitting at a tiny 0.5% usage rate, suddenly saw a tenfold surge in usage.
Just because they physically moved closer to the action.
Exactly. The researchers mapped this out visually using a graph called a principal component analysis, or PCA.
Okay, how does that look?
Imagine a graph where each dot represents the total immune profile of one single mouse. The control mice, whose DNA wasn't shuffled, all clustered tightly together in one tiny corner of the graph. Their immune profiles were nearly identical.
But the mice that received the chemical trigger, the ones that got the plant hormone,
Their dots were wildly dispersed across the entire graph. They were totally different from the control mice and completely different from each other. The shuffling completely destroyed the rigid natural rules of the DNA, massively expanding the antibody diversity.
So let's recap this final amazing piece. They chemically forced the DNA to generate entirely new immune patterns. We started with a rigid, linear system where one piece of DNA could only do things one way. And by pausing the system and adding a chemical trigger, they broke the hardware open, allowing it to rapidly generate massive, unpredictable diversity.
It transformed a rigid one-to-many process into an explosive many-to-many framework.
Which really makes you wonder, as a listener, if your own cells could dynamically scramble their DNA on command to solve an incoming threat, what limits, if any, should we place on that kind of biological unpredictability? It fundamentally changes the capacity of a living organism to adapt.
It does. And if we connect this to the bigger picture, the implications of this go far, far beyond just immune cells or antibodies.
How so?
What Fu and his team have established here is a scalable, generalizable framework to design, build, test, and learn the architecture of literally any mammalian genome. If we can build a vacuum sealer protein to safely assemble massive strings of hyper-repetitive DNA, and we can make that DNA dynamically rewrite itself inside a living mammal, we now have a systematic way to rewrite complex disease genetics anywhere in the body.
So we're officially moving from merely observing biology to fully programming it.
Exactly. We're writing the software of life now.
It's an incredible paradigm shift. Thank you to you, the listener, for coming with us on this dense, fascinating journey. We started with the physical limitations of an ancient biological library, navigated through crowdsourced synthetic blueprints and DNA compacting vacuum sealer proteins, and arrived at living mammals that can literally shuffle their own immune code with a chemical switch.
It's been quite the ride.
It really has. But before we go, I want to leave you with one final provocative thought to mull over. We opened by talking about how your natural immune system builds blueprints to fight novel viruses based on a static set of rules. But if we can now engineer synthetic immune systems that can learn, invert, and shuffle their own genetic code on a specific chemical command, could we eventually pre-program human cells to continuously generate novel defenses against future, entirely unknown pandemics? With a self-shuffling synthetic immune system waiting inside us, constantly generating new armor on the fly, could the concept of a traditional vaccine eventually become completely obsolete?
Now that is something to think about.
Until next time, keep diving deep.